Okay, so welcome back to this next video in which we are discussing the antithrombotic mechanisms of the endothelium. Okay, so we're in the process of revising the hemostatic pathway now. Okay, so what happens first? Let's revise what we've done so far. What happens first is that the damaged endothelium has exposed von Willebrand factor, which can then bind to the glycoprotein 1b95 on the surface of the platelets, okay? So you're going to get platelets adhering to this site of damage here, okay? Or at least to the mouth of the site of damage. So all around the hole in the endothelial cells, you're going to get platelets adhering there, okay? Then some other platelets are going to go into through the hole in the endothelial cells into the deeper layers of the wall of the blood vessel, and they're going to be activated by tissue factor and by collagen, okay? And when they're activated, what that means is they release ADP and thromboxane A2, and I want to stress this again, that platelet activation means that the platelet has started to release ADP and thromboxane A2. That's the process of activation. Okay, now the ADP then triggers a chain reaction by binding to ADP receptors on other platelets and triggering those platelets to undergo activation and start releasing ADP and thromboxane A2. And this results in all the platelets in the vicinity of this hole in the wall of the blood vessel releasing thromboxane A2. So you get massive levels of thromboxane A2 in the vicinity of this hole. That leads to the contraction of the smooth muscle cells lining the blood vessel nearby this hole, and that's going to lead to vasoconstriction to stop blood coming through the lumen to where this hole in the blood vessel is. Okay, so the other thing that thromboxane A2 does is it binds to the thromboxane A2 receptor on all of these platelets here, and is going to lead to them becoming sticky. So it activates the GP2B/2A protein, which means that they can now start to stick together. Okay, so if we put everything together, we've got the activation going on, which it leads to the production of the thromboxane A2. Thromboxane A2 then makes the platelets sticky by activating GP2B/2A, and now what's going to happen is that the platelets are going to start to stick together. Now we've already got these platelets which are adhered to the mouth of the wound, okay? So I might draw another picture of this over here. Okay, so if we've got the mouth of the wound here, the endothelial cells which line this hole, okay, we know that we've already got platelets which have adhered to these damaged endothelial cells here, and they've adhered by binding to the von Willebrand factor which they are now exposed to. Okay, so these are platelets here. So then let's put some more layers on here. So these are platelets which I'll just highlight in orange. Okay, but it's going to go a horrible colour because it's so small. Okay, so those are the platelets, and then underneath the endothelial cells we know we've got the broken basement membrane here. Okay, and then underneath the basement membrane we've then got the subendothelial connective tissue here, and then finally the internal elastic lamina. This is the picture that we had before. Okay, so here's the internal elastic lamina, and then the smooth muscle cells will be around that. Now what's going to happen is all the platelets are going to get sticky, and they're going to start the other platelets, you know, which have undergone the activation process to release thromboxane A2, and then thromboxane A2 has made them sticky by activating the GP2B/2A. They are now going to stick to these platelets which underwent the initial adhesion process, okay? And these platelets that underwent the initial adhesion process, they've also had thromboxane A2 slobbered over them, okay? So they also have activated GP2B/2A proteins, so then fibrinogen can be mounted in between two GP2B/2A proteins on different platelets, and you can get the formation of a platelet aggregate. And to get the point across, I think I'll just draw the platelet aggregate happening to stretch right over the mouth of this hole here. Okay, so there is our platelet aggregate, and you can see how now this is going to plug the hole in the blood vessel wall. Okay, so this is our platelet aggregate. Okay, now this is not the full plug, basically. You're going to have a stronger plug than this because these interactions between platelets aren't actually brilliantly strong. This is sort of like a very gooey plug that would break very easily, basically. Okay, so it has a name. It's got a name. This sort of a plug that plugs the hole in the blood vessel wall. This is known as a primary platelet plug, or it's also referred to as a primary hemostatic plug. So primary platelet plug is the name that often people use for this, but the more correct name is to call it a primary hemostatic plug. And again, I'll use the British English spelling of hemostatic just because it's more natural to me. Hemostatic plug. Okay, right. So this formation of the primary hemostatic plug, this is the second portion of the hemostatic response. Now let's move on to the third portion of the hemostatic response. So the third portion of the hemostatic response is coagulation. Okay, so we need to discuss what coagulation actually is. So, coagulation then. Basically, in the blood, there is a protein that we've already seen the name of. We've already seen fibrinogen. Okay, so this is this protein in the blood, which is involved in aggregating the platelets. It's involved in binding to two separate GP2B/2A proteins on two separate platelets and leading to this aggregation process here and the formation of the primary platelet plug. However, fibrinogen has another role in this hemostatic process, so it's very, very important protein. So fibrinogen, or factor 1 if you so wish, is produced in the liver and it's usually circulating in the blood, okay? Now, if you've got no holes in the side of your blood vessels, then fibrinogen just goes around, all it sees is the other components of the blood, and it only sees the endothelial cells. So it never gets activated. However, it can be activated to another form, to the active protein. It can be turned into a protein known as fibrin, and this, in the old clotting factor notation, would be called factor 1 with a little a at the bottom to denote that this is the active form of factor 1. Okay, now the active form of factor 1 can then be assembled into great big fibrin strands. So you can polymerize fibrin into massive, great fibrin strands that are actually, they're big structures. They're structures that you can easily see down a microscope, okay? Whereas obviously you won't be able to, down a light microscope, so let me stress that. Obviously you wouldn't be able to see the protein, the individual fibrin protein down a microscope, certainly not a light microscope anyway, maybe down a transmission electron microscope, I don't know. But these fibrin strands can be seen down the light microscopes, they're big. Okay, so coagulation is the process of taking this inert, tiny little protein which is just circulating in the blood and turning it into these fibers, basically. Now, what's the function of these fibers? Well, basically they are going to hugely strengthen this primary hemostatic plug. So this coagulation process, we are going to activate it. It's going to be activated again by the same things which activate this formation of the platelet aggregate. So it's going to be activated by collagen and by tissue factor. So it's going to be activated in the same place as we are building this primary hemostatic plug. So you're in amongst all these platelets which are aggregating together, you are going to get coagulation occurring. You're going to get fibrinogen being converted into fibrin, and then fibrin being built into fibrin strands. And what it will produce basically is an intertwining mesh of fibrin strands which will intertwine amongst all of these platelets here that are forming this primary hemostatic plug, and will basically form like a spider's web which intertwines in every little nook and cranny basically, and is sort of holding every single platelet together, because all of the platelets have their position within this spider's web. So you have a meshwork that surrounds all of these platelets and is holding them together. So let me emphasize this still further. So in these red lines represent the fibrin strands, okay? So they will intertwine amongst every single platelet, and you won't just have one fibrin strand in each of these places. You'll have many, many, forming a really dense spider's web-like meshwork that surrounds each and every platelet and holds each platelet to the neighbouring one. And this fibrin meshwork really is what holds together the hemostatic plug. And when you convert a primary hemostatic plug, which is just the mush of platelets all aggregated together, to this mush with a very strong mesh work around it. The structure with the very strong mesh work around it is what's known as a secondary hemostatic plug. Okay, and these secondary hemostatic plugs are far stronger than the primary hemostatic plugs. So the fibrin meshwork around the platelets is essential for creating a strong, viable plug in this hole in the blood vessel. Okay, and we'll discuss the process by which you activate the conversion of fibrinogen into fibrin in the next video.